Wishing you all a very good morning on this auspicious day. With the blessings of Divine Holiness, Param Pujya Hari Prasad Swamiji and Param Pujya Prem Swarup Swamiji, I, Dr. Hitarth Bhatt, feel privileged to welcome you all in the inauguration program of DBT and CSIR sponsored National Conference on Environmental Microbiology and Regulatory Aspects, which is organized by Atmiya University and Shree and Virani Science College, autonomous affiliated to Saurashtra University. It is my honor and privilege to stand before you today and as we gather to explore and discuss the intricate relationship between microbiology and our environment, along with the critical regulatory aspects and framework that shape our actions and decisions in the field. This conference is a fantastic opportunity for us to come together as a community and share our knowledge and expertise on this a very vital field of research. We have a lot to learn from each other, and we hope that this conference will provide a platform to do just that. I request the conveners of this conference to please escort our guests and dignitaries on the dais. May I request our esteemed speakers, Professor Hemant Purohit, former head, Environmental Biotechnology and Genomics Division, CSIR Neri, Nagpur, Professor Dayanand Naxar, Vice Chancellor, Gulbarga University, retired Senior Professor Sasikala, Jawaharlal Nehru Technological University, Hyderabad, Professor Arun Kharat, School of Life Sciences, Jawaharlal Nehru University, New Delhi, Dr. Madhvi Joshi, Joint Director, Gujarat Biotechnology Research Center, Gandhinagar, Dr. D.D. Vyas, Registrar of Atmiya University, Dr. Ashish Kothari, Director, Research, Innovation and Translation, Dr. Nipa Pandi, Head of the Department, Shree and Virani Science College, and Dr. Rohan Pandya. I also request Professor Samir Vaidya sir to please come on the dais. Please, sir. As per our culture and Indian tradition, we always start any occasion by first remembering and worshipping God. I request audio team to please start the prayer. this line, let us first take blessings of Thakurji. And for that, I request Dr. Rohan Pandya, Head of the Department of Microbiology, Atmiya University, to please garland to Thakurji. Thank you, sir. Now I request our Honorable Registrar, Sir, Dr. D.D. Vyas, to extend floral welcome to Professor Samir Vaidya, Secretary, Sarvodaya Kelavani Samaj. Thank you very much, sir. Now I request Professor Samir Vaidya, sir, to welcome and felicitate our today's keynote speaker, Professor Hemant Purohit, former head, Environmental Biotechnology and Genomics Division, CSIR Neri, Nagpur. Please, sir. Thank you very much, sir. Now I request Dr. D.D. Vyas, sir, Registrar of Atmiya University, to welcome and felicitate Professor Dayanand Akzar, sir, Vice Chancellor, Gulbarga University, Karnataka. Please, sir. Thank you, sir. Now I request Dr. Neepa Pandey, Head, Department of Microbiology, Shree MNN Virani Science College, to welcome and felicitate Senior Professor Sasikala, Jawaharlal Nehru Technological University, Hyderabad. Thank you, madam. Now I request Dr. Rohan Pandya, sir, to welcome Professor Arun Kharat, sir, School of Life Science, Jawaharlal Nehru University, New Delhi. Thank you very much, sir. Now I request again Dr. Neepa Pandey to welcome and felicitate Dr. Madhvi Joshi, Joint Director, Gujarat Biotechnology Research Center, Gandhinagar. I request Honorable Registrar Sir Dr. D.D. Vyas to extend floral welcome to Professor Samir Vaidya, Secretary, Sarvodaya Kelavani Samaj Sir. Okay. We are very happy to have with us Mrs. Hemant Purohit and Mrs. Dayanand Akhzar. We wish to felicitate you as a token of our love. I request Dr. Raksha Bhavankar to felicitate Mrs. Hemant Purohit with flowers. Thank you, madam. Now I request Dr. Krishna Joshi, madam, to welcome and felicitate Mrs. Dayanand Akhzar. Please, madam. Thank you very much. Thank you all. Now I request all the dignitaries to come forward for lighting the lamp, which signifies the beginning of this conference, and pray for its success. Please, I request conveners of this conference to please escort the guests for lamp lighting. Thank you all. In recent years, the significance of environmental microbiology has become increasingly evident. Microorganisms play a fundamental role in shaping the health and balance of our ecosystems, influencing everything from nutrient cycling to pollutant degradation. And as we confront pressing environmental challenges such as climate change, pollution, and emerging infectious diseases, understanding the role of microorganisms becomes paramount. This conference brings together experts and researchers from various fields of environmental microbiology to share their knowledge, research findings, explore new horizons, and to build network that will foster collaborations among the scientific community. Without much delay, I request Dr. Rohan Pandya, convener of this conference, to give welcome address. Please, sir.
Namaste and Jai Swaminarayan to all. Before we proceed further, it is essential to acknowledge the omnipresence of the divine guidance of Brahm Swarup Guru Hari Prasad Swamiji Maharaj and Guru Hari Prem Swarup Swamiji Maharaj, who have provided unwavering support and guidance to all of us. It is also necessary to acknowledge the mentoring provided by our beloved mentor, Param Pujya Tyagvallabh Swamiji Maharaj, who has always guided us for the best of our efforts. I also like to acknowledge the guidance provided by our Professor Samir Vaidya sir for joining the session and provide necessary advice to shape this particular event. I take this privilege to welcome you all in this inaugural session of the DBT and CSIR supported National Conference on Environmental Microbiology and Regulatory Aspects, hosted by Department of Microbiology, Atmiya University and Shree MN Virani Science College, Rajkot. We are immensely delighted to have you all here as we embark on the 2 days of the insightful discussion and knowledge exchange. We are honored to have a distinguished speaker on the dais with us today who are expert in their field, and we are very eminent persons of that respective areas. Today we welcome our keynote speaker, Professor Hemant Purohit sir, and along with Professor Dayanand Akhzar, Professor Shashikala, Professor Arun Kharat, and Dr. Madhvi Joshi, who will be sharing their valuable insight and experience with us today as well as in tomorrow's sessions. We are truly privileged to have such eminent personality contributing to the success of this particular conference. I would also like to welcome Dr. D.D. Vyas, Registrar, Atmiya University, and our organizing secretary, Dr. Abhijit Joshi and Dr. Raksha Bhavankar, who has provided very best support to organize this particular session. I also like to thank you, our co-organizer, co-convener, Dr. Nipa Pandhi Madam, who has also constantly provided necessary support and all the advice to shape this particular event. At the beginning, this is an inaugural session, so I also would like to welcome all the dignitaries who has joined of the dais, deans of the faculties, head of the departments, sponsoring partners, faculties from the various departments, scientists, academicians, research scholars, and all the students, participants in this particular conference. I wish welcome. I hope that this particular 2 days interaction of the conference will be very much fruitful and shaping a much more brighter path in our career journey. Thank you. Thank you very much.
Thank you very much, sir. Now I request our registrar, Dr. D.D. Vyas, sir, to please read presidential address and enlighten all the participants. Please, sir.
Thank you. With humble gratitudes in the lotus feet of Brahmaswarup, His Divine Holiness Param Pujya Hari Prasad Swamiji Maharaj, Guru Hari Param Pujya Prem Swarup Swamiji Maharaj, our guide, our mentor, and President of Atmiya University, Param Pujya Tyagvallabh Swamiji, respected dignitaries on the dais, Dr. Samir Vaidya sir, Dr. Professor Hemantji, Professor Dayanandji, Professor Shashikala Madam, Professor Arunji, and Dr. Madhvi Joshi ji. On behalf of Atmiya University, I join my colleague Dr. Rohan Pandya to welcome all dignitaries, participants, head of the departments, faculties of the Dean in this inaugural session of 2 days National Conference on Environmental Microbiology and Regulatory Aspect. I would like to express my sincere thanks to all the experts who have given their consent and are present today and the day after for various expert sessions, plenary sessions where they would be sharing insights into the trends and the importance of the very topic on the conference. I would also like to congratulate the team of Department of Microbiology both at Atmiya University, as well as our sister concern, Shree and Virani Science College, for planning this conference. I understand that this is the second conference that they have planned in series, one in 2023 and this in 2024. I appreciate the efforts that had been put in by the team led by Rohan sir in meticulously designing this program, this conference, inviting experts and trying to take every measures so as to make this conference productive. As has been mentioned by the host of this today's inauguration function, that because of some prior commitments, our president of the university, Tyag Swamiji, could not be with us today in this function. However, Swamiji has sent his address and blessings for all of us, and I do have this honor to read that in front of you. I quote, J. Swaminarayan, dear participants, I would like to express my sincere welcome to all the prominent attendee, researchers, academics, academicians and guests to this National Conference on Environmental Microbiology and Regulatory Aspects, organized on 23rd and 24th of February 2024 at the campus of Atmiya University. We are excited to be hosting this important conference, which provides a forum for the sharing of insights, innovations, and discoveries in the areas of regulatory issues and environmental microbiology. Our dedication to encouraging thought-provoking discourse and research in this important field is consistent with the objective of making a positive impact on a healthier and more sustainable planet. The conference offers a singular chance to convene specialists, scholars and industry professionals from a range of backgrounds to deliberate on the most recent discoveries, breakthroughs and legal frameworks that influence our understanding of environmental microbiology. This event is expected as a hub for cooperation and information sharing as we work together to improve our comprehension on how microbes interact with the environment and also how the legal system is managed related to that. I urge every person to participate fully in the sessions, contribute their knowledge, and develop deep relationship with other professionals in the industry. By working together, we can open the doors to a more knowledgeable and practical approach to regulatory compliance and environmental microbiology. On behalf of Atmiya University and Shree M. Virani Science College, I want to thank the sponsors, the organizing committee, and everyone else who helped to make this conference possible. Your commitment to furthering scientific understanding is incredibly admirable, and I am confident that this event will be a huge success. Many revolutionary discoveries and cooperative efforts towards a sustainable and peaceful coexistence with our environment be sparked by this National Conference on Environmental Microbiology and Regulatory Aspects. Wishing a productive and enriching conference. Jai Swaminarayan to all. With this words from Param Pujya Swamiji, I wish all the participants a very best in this conference. Thank you very much.
Thank you very much, sir, and thank you, Swamiji, for your blessings. Now I request Professor Samir Vaidya, sir, Secretary, Sarvodaya Kelavani Samaj, to address this gathering. Please, sir.
Good morning and Jai Swaminarayan to all of you. Dignitaries sitting on the dais and dear students. It is my great privilege to welcome you all on the campus of Atmiya University and MNN Virani Science College. As our registrar, Dr. D.D. Vyas mentioned, that this Atmiya University is a non-profitable and non-commercial university, which is always worrying about the holistic development of students. Recently, in National Educational Policy 2020, which specially emphasized on value education. But it is a great honor to mention that this university has worried about the value education since 2018. And I am also very much proud to mention that we are the front runner to think and include the value education as a part of our curriculum. And it is due to the blessings of the founder president Hariprasad Swamiji and his successor Prem Swarup Swamiji and the president of Tyagvallabh Swamiji. Because they believe that the education without value is of not a value. And that is why the university is always worrying about the advanced curriculum. All the faculties and the management of Atmiya University is very much proud to welcome you all on this campus. And the theme of this conference is also very much relevant to discuss the contemporary issues, the environmental biology and regulatory aspects. I am the man of mathematics, but I first time encountered the frontier between the microbiology and mathematics. And our common experience is that if we are suffering with even common cold and we approach to our family physician, immediately they ask that and prescribe for TC, DC, ESR. Then once with the curious nature, I asked my family physician, what is DC? Then it is differential count, total count. And then it is the first frontier and the connection and interaction between the two emerging fields of science, that is mathematics and microbiology. So that is the thing, that now we cannot work in silos. We have to work in an interdisciplinary, in particular, and in transdisciplinary way in general. And I think this aim will be naturally fulfilled during this conference. So again, I welcome you all and wish a grand success to this national conference. Thank you.
Thank you very much, sir, for your motivation. Without further ado, I am delighted to announce the official release of the abstract book. All the dignitaries are requested to officially release this abstract book of this 2 days national conference on environmental microbiology and regulatory aspects. Please round of applause. Thank you all. Thank you very much. Now I call upon Dr. Neepa Pandey, Head of the Department of Microbiology, Shree and Virani Science College, and also one of the convener of this conference, to deliver vote of thanks for this session. Please, Madam.
Thank you, Hitarth. Good morning and Jai Swaminarayan to all. Appreciation is a wonderful thing. It makes what is excellent in others belong to us as well. This is said by Voltaire, a French environment writer. And in the quest of harassing and harboring the excellence accumulated in this arena, I humbly take this privilege of extending vote of thanks. On behalf of the entire organizing committee of this National Conference on Environmental Microbiology and Regulatory Aspects, I am indebted to Brahm Swarup Param Pujya Hariprasad Swamiji Maharaj, Founder President Yogi Divine Society, Haridham Sokhda, and Param Pujya Prem Swarup Swamiji Maharaj, President SKS, for their divine blessings which continuously empower us to deliver our best in all our endeavors. Please continue to shower your choicest blessings on us, Swamiji. I offer heartfelt gratitude on the lotus feet of Param Pujya Tyagvallabh Swamiji Maharaj, Founder President Atmiya University and Secretary Sarodai Kedavni Samaj, for his loving guidance, care and constant motivation to go beyond the boundaries and to deliver the best. He is the pillar of strength, supporting every initiative taken on this campus. Kindly accept our pranam, Swamiji. I am indebted to Professor Swamir Vaidya sir, who is Secretary SKS, for sparing his time and to be present here and bless us with his encouragement and blessings. Thank you, sir. It is with great joy and pride that I acknowledge the presence of our distinguished guests and speakers, Professor Hemant Purohit, Professor Dayanand Aksar, Dr. Arun Kharat, Professor Sashikala, and Dr. Madhvi Joshi in this inaugural session. Thank you, sir, madam, for sparing your valuable time. We are indeed privileged to have highly distinguished and extremely effective leadership team bearing torch to the path of accomplishments. I extend gratitude to Professor Sheila Ramachandran, Pro-Chancellor, Atmiya University, Professor Shiv Tripathi, Vice-Chancellor, Atmiya University, Rajkot, the Pro-Vice-Chancellor of Atmiya University, Dr. Jayesh Deshkar, Principal Dr. K.D. Ladwa at Virani Science College, Rajkot, Dr. D.D. Vyas, Registrar, Atmiya University, and Dr. Ashish Kothari, Director of Research, Innovation and Translation, for their continuous support, continuous encouragement, motivation, and providing every single facility that we ever required in the form of guidance or infrastructure to make this conference possible. We are indeed very much thankful to you, sirs and madam. And I think I would be very much unfair if I don't thank all the online and offline participants who have been here, present here, for sharing their knowledge, gaining of the knowledge, and to make this conference a real success. Thank you all. Thank you very much. Thank you.
Thank you, madam. Now I request all the dignitaries to gather for a group photo on this stage. Please, sir. Stage. 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 Thank you all. Moving forward, now it is time for a keynote address. I request all the dignitaries to take their seats in auditorium of the dais, and I request Dr. Rohan Pandya to kindly escort the guests. सर इधर सब व्यवस्था है, आपको बस पेन ड्राइव दे देना है. They will operate the laptop for you. You can say next. They will. Okay, we have also pointer. Okay. So whatever you wish. You want pointer? Yeah. Okay. Fine. फटाफट. माइक कराइए. પેલા ગોતી લેવા દે, પછી પોઈન્ટર ગોતી લેતું સર. It is my privilege to introduce our keynote speaker, a renowned scientist, Dr. Hemant Purohit, former head, Environmental Biotechnology and Genomics Division, National Environmental Engineering Research Institute, that is NEERI, Nagpur. Dr. Purohit has published more than 252 research publications in international journals of repute. He has also published 19 book chapters, and he is the author of 4 books. He has 3 Indian patents, 2 USA patents, and 2 WTO patents to his credit. He has mentored 22 doctoral students. He has completed several major research projects from various funding agencies, and he is a member of prestigious national and international societies. Dr. Purohit is also serving as a member of different task forces of DBT, Government of India, and Senate member of RTM, Nagpur University. Dr. Purohit is a recipient of a very prestigious award such as Fogarty Fellow and Associate at National Institute of Neurological Disorders and Stroke, NIH, USA.
Thank you.
And he was also a Commonwealth postdoctoral fellow at Department of Biochemistry, University of Hull, UK. Dr. Purohit is a member of editorial board of different reputed journals such as PLOS One, Applied Biochemistry and Biotechnology, Springer, and Indian Journal of Microbiology, Springer. He is going to deliver a keynote address on managing health challenges of the ecosystem. I request Dr. Purohit to kindly come on the stage for keynote talk and enlighten participants with your knowledge. Please, sir.
Thank you. Hello, am I audible? Yes. Thank you. Chalo, Jai Swaminarayan. So, at the outset, I am very thankful to the organizers to bring me here and talk to all of you. Actually, when I did my PhD, I couldn't attend my convocation. And finally, I got my convocation done today. So let's talk about managing the health challenges of ecosystem. Are you sure this is working? Yes. Okay, fine. So let's talk about managing the health challenges of the ecosystem. And when we say the challenges in the ecosystem, first we should understand what ecosystem is that and why we should be managing the our. हो गया भाई चलो so when we are managing our ecosystem, essentially we talk about managing our resources, and our resources are essentially the animal resource, solar energy, water resource, forest resource, coal resource, soil resource, and oil resources. And these resources, if they are managed properly, then we are talking about we are doing the ecosystem services. So in that case, we manage because we want to have our environment protected, we want to have our social progress, we want our economic development. And these three things are linked with our sustainable development. If you are looking at the broad range of sustainable development growths. Now let's talk about in terms of broadly first about the ecosystem. And then gradually we focus on to the different issues. So if you are looking at an ecosystem and we are looking for the energy flow, the main source of energy, everybody knows that we get a solar energy, which is getting fixed by the producers. So there the inorganic small nutrient pool is getting converted into the organic biopolymers, and these organic biopolymers are finally consumed, and finally at that cost, this whole ecosystem survives, and finally the degraded products are coming into back to the pool through the decomposers. So this is the way the energy flow of the system works. And when we are looking for the energy flow and when we link to the food web, then we realize that the sun, and then finally we know the plant as the first biosynthesis machinery which works in the ecosystem and converts the solar energy and the micronutrient pool into the different food webs and serves to the different food webs. Then when we are talking about now food webs, so we have different producers, consumers, and finally the decomposers. And that way our food web basically works. For different ecosystems, we have different kind of food web systems. But primary source of energy remains same. Primary decomposers remain same. The bacteria and the fungi, they become the primary decomposers. And then we are having the marine food web. So that way for every ecological niche, we can define the food web. And these food webs essentially create a micro niche for every system. And in that micro niche, finally the products are getting decomposed and then they are bringing back to the ecosystem. So that's the way the pool of energy or pool of nutrient essentially flows in the ecosystem. When we talk about the nutrient pool or inorganic nutrient pool, in presence of energy, they are converted into carbon cycle, nitrogen cycle, and the phosphorus cycle. So all these cycles essentially decides in what manner the ecological resources should be recycled, reused, and essentially made available to the main consumers. And we as a human, with our selfish attitude, we try to consume as much as possible. And that has to be basically balanced. So when we talk about ecosystem per se now, we have an atmosphere, we have a lithosphere, and we have a hydrosphere. And these three together becomes biosphere. So now if we are talking about management of ecosystem services or ecosystem health, then any perturbances or any developmental activity means we are talking about industries, we are talking about any kind of development, these perturbances leads to the challenges to the ecosystem. And our atmosphere or biosphere or lithosphere or hydrospheres get perturbed. Even suppose we come with an industry, then this industry essentially puts stress on the land ecosystem, even as the water ecosystem, and finally at the air or atmosphere ecosystems. So once we have a stress on biosphere, it exerts the stress on society and finally the economy. So if we manage our biosphere right, if we manage our health of the biosphere right, then definitely we can manage the society and economy. So for that kind of things, what as a society we require? As a society we require policy, incentives and regulations. If they are in a place, or if we want to create this in a place, then we require a sustainability design, that we have to decide in what manner, which part of the lithosphere or biosphere we want to sustain. Hey, don't worry. It's there. Then sustainability, and then with that we collect the data. With the data we develop the complex model. Why I am saying the model complexity? Because we are having different attributes at different levels. And these attributes directly and indirectly react with each other. And these interactions basically decides what kind of sustainability indicators will emerge over a period of time. These sustainability impacts, they get impacted, and accordingly we have to design these changes in our policy or incentives. What I mean by that, if suppose our model says these are my sustainability indicators, and if we look at the impact and we think that this is not working for us, then definitely we have to look for technology improvement or different way of designing the sustainability parameters. So that way the regulations are made, that's the way the systems will be evolving with time. Then if we are looking for the integrated parameters to develop the sustainability, then we have to talk about important parameters are the environment. If the environment is right, the social parameters gets in place, and for that we have economics, ethics and governance supporting them. So if we are talking in principle, the sustainability of environmental system, then for defining the ecosystem health, we have to have environmental health in place, soil health in place, and One Health in the place. One Health is now the emerging parameter which is coming up with the COVID and other pandemics which are coming, right? And with that we are now getting aware of this kind of ecosystems which are emerging with time. And if we manage them right, then we are managing the healthy people, the healthy environment and the healthy animals. So let's talk about now One Health, soil health and environment health in separate. And let's see how this could be managed and what are the different challenges there. So first let's talk about One Health challenges. When we are saying One Health, essentially we are talking about human health, animal and plant health. The ecosystem health is directly related with the food security, and it is also related with the social well-being. Then now we have to understand how the ecosystem health is related with the One Health system. When we are saying ecosystem health and One Health, then in humans, in wildlife and livestock, through various pathogens we are linked with each other. Either directly human wildlife or human livestock, or we are having some common pathogen which are working across the three ecosystems or ecological niches. What really happens over a period of time, a pathogen really evolves, finds a new host, and for its survival, after looking for the new host, it acquires the new characteristic. And these new genetic characteristics essentially poses a problem. And these problems are expressed in different models in the different way. And that is the biggest challenge. It is not like the same pathogen will be expressing their properties in these three ecosystems in the similar manner. So what is the evolving scenario in One Health? If we are looking at the scenario evolving in the One Health, we can see the population growth, healthcare system, and mobility and transport. They are the new drivers which is putting the pressure onto the human ecosystem, because we are linked with the mass production, urbanization, deforestation, and travel and tourism. This travel and tourism, deforestation, and urbanization, they are the drivers for environment. And once the environment is getting destabilized, then it is indirectly putting the pressure onto the human system. Another thing is the driver is the population growth, as I said. The population growth demands food and different farming practices to meet the food demands. According, the international trades are getting affected. Once they get affected, the animals are getting affected. So this is the way, the new way of movement of different pathogen across the different ecosystem is now getting studied, and this is the evolving scenario here. Immediately, once we put this thing in context with the One Health, what is really coming up, the new identified problems are zoonotic diseases, antibiotic resistance, food safety and security, waterborne diseases, environment health, chronic diseases, mental health, and occupational health. These are immediately been identified after looking at these interworking scenarios. And these are essentially part of all the three ecological niches. Now, in this, the identified problems, we have stressors, reservoirs, process, and health mediators. Stressors means, let us say, if suppose you are having agriculture and you are using a lot of pesticide. So once you put a lot of pesticide, that becomes a stress in the ecosystem. That creates the different kind of pressure and develops the different kind of reservoirs. What I mean by reservoirs, that you are having, suppose, antioxidant pool, you are having free radical pools coming up. And these free radical pools creates a process in which you are having either the antibiotic resistance emerging or you are having cancer emerging. So these kind of processes are coming, and finally you get different kinds of health mediators. So to meet these problems, there are different programs. Initially, these programs are like zoonotic diseases for this, that, and so on. But now these programs are becoming very generalized programs. They are not very specific programs. The reason is that things are very interrelated. So therefore, in One Health issue, there is a EU program came up which says Versatile Emerging Infectious Disease Observatory. This program essentially takes care of, in different 19 countries of EU, they want to figure out how to bring up with early warning, how to do the risk assessment, monitoring of emerging and infectious diseases, and then also monitoring the antimicrobial resistance, in what manner it is coming up. Right? So this is, they are talking in terms of EU program, and in that, the idea is that in this versatile emerging infectious disease, or this vehicle, they are talking about, essentially before pandemic, we were talking about only this current approach, that how humans are getting affected. Now they are talking about in terms of how the wildlife linked with vectors, linked with the domestic animals, and how this is getting transferred to humans. So you are talking about in what manner a particular genetic determinants gets spillover in the different ecosystems, and while it is getting spillover, this genetic determinants, how it getting evolves. Right? In what manner their different genes interact with each other and manifest different kind of phenotype or different kind of etiology to the ecosystem. So this is what they want to study here. If we summarize, there is another program which is a Netherlands program. EU program essentially is oriented with reference to different etiological agents. So we are talking about different viruses, pathogens. Then viruses, pathogens means genetic determinants. Then AMR. Then we are talking about different pathologies, right? But when we talk about Netherlands program, they are more concerned about the climate change, water management, farming methods, and international travel and import risk. So Netherlands program essentially is a behavioral program in what manner we are managing the different part of the environment rather than we are managing the different types of pathogens. So these are the different programs which are coming up. There are various programs, but I selected these two for the discussion because one basically talks about different pathogens and how these pathogens are evolving, and another is the different drivers which is deciding in what manner the etiology will evolve or could evolve. Here we require a lot of mathematical modeling to assess or to figure out or predict in what manner the different models are getting emerging. So in the context of One Health now, the strategies are changing. The emerging strategy says, first develop the program, then identify the network. Network is very important in these ecosystems. Network means even a farmer, a cattle, human being, water body, air quality, migratory bird, all they become the networks to bring a typical kind of a pathogen into your ecosystem. Then map that, analyze that, and then look for the opportunity in what manner you can manage that. Because like pandemic told us that quarantine, or let us say you shutdowns, this is not finally the answer for sustainable development. So we have to have proper mapping of different intertwining networks and how we can disrupt these networks so that way there is no flow of basically the pathogen. That has to be decided. So if we reframe this strategy, then it takes that you have a zoonotic pathogen, going to the wildlife, going to the ecosystem. From there the outbreak comes. You have domestic animal, humans, wildlife. You already know all this part. But what the suggestion is now, let us say if I have a COVID type of a primer with me. So if I am doing RT-PCR and I am saying that, okay, humans are now still having that, or if I am monitoring the sewage and if I am saying, even if I can see now the genetic determinant for a particular virus, that means this pathogen is surviving. Instead of that, they are saying that we should randomly taking the sampling all the time. And with that, we should always be looking for new determinants or gene coming up into the ecosystem. That is the whole idea of the program. So if this strategy really works out, then finally they are saying the early detection of pandemic virus, early warning and early eradication. So if we do that finally, then we don't have the problems of One Health coming up. That is the idea. Now if suppose now we take this further up, how the One Health linkages basically developing over a period of time. So One Health linkage, if you have a healthy human, healthy planet if you want, then this healthy soil is the key. That's the environment part, which is very interesting and very much emerging now. And they are linking every single thing, every single disease, every single pathogen, or every single, you can say, see in the language of genomics, now pathogen is no relevance now. Now the pathogens are basically carrier of a specific genes. And what essentially brings the disease is the genes or the interaction of genes. A particular gene could be a silent gene in a particular host, but it can be a very aggressive gene in a different host. So the environment of gene with reference to different ecosystem and stress of that ecosystem of this gene really expresses in a different manner. So that essentially decides in what manner the disease will evolve or will move. So let's talk about now soil. And this is almost 15 years back. William Albert has written that it is high time we learn that our national health lies in our soil. So they are linking soil with the health. Why? Because when the fertilizer has started, when the pesticide has started, they say that if you won't save soil, you cannot save our health. So let's now focus on soil health with reference to the managing the ecosystem and challenges of the ecosystem. So what soil health is and why soil health we should be worrying about? Because soil is directly linked with the livestock, through the agriculture, through the drinking water, through the lakes, through the various events, we are essentially linked with the soil. Soil is linked with the plants, plants linked with the livestock, linked with the antibiotic microbial resistance marker, and then whole food web indirectly is linked through the soil. So if you're just looking at soil and livestock and human, you think in terms of a food web. And then you will find that yes, soil has a very big relevance with reference to the movement of different markers. If you look at the soil and the different linkages across atmosphere, lithosphere, anthrosphere and everything, then it has a lot to do with human health, well-being, and it is then linked with the ecosystem health. So soil health becomes very important if you are worrying about ecosystem health. Because what soil does? Soil essentially provides the soil function. Soil function means all your biogeochemical cycles, they are basically decided by soil. If these cycles are right, then the ecosystem services are really good. What I mean by that, if your nitrogen fixation is right, carbon fixation is right, then definitely the plant growth will be right. If the plant growth is right, the whole diversity is supported properly, from bacteria to insect to different birds. So those comes under ecosystem services. And if this is all linked, then finally you link with the sustainable development goals. So soil health becomes very, very crucial in that way. Then again, I'm bringing back the same slide that ecosystem health is has linkages with the food security. So now let's link soil health with the food security. So that way we can talk about social well-being also. And that way indirectly we are talking about managing the environmental or ecological health. So in soil health, the biggest drivers are climate change, nitrogen depositions, invasive species and pollution. Climate change we cannot directly control, but indirectly we are contributing in terms of deforestation. When it comes to nitrogen deposition, means we are excessive use of fertilizers. Then come invasive species. You know that nowadays people are doing agroforestry where you have a single type of plants, only doing the plantation. So once that comes, the biodiversity loss is very huge. And then comes the pollution in terms of what kind of chemicals you are putting into the ecosystem. So because of that intensive land use management, we are losing diversity, we are increasing pest and pathogen, and beneficial organisms are basically we are wiping out from the ecosystem. And that way, we have to, if we don't shift slowly to sustainable agriculture, then this problem will become very significant. If we look at the long term in terms of food security, then what happens? Our driver becomes land use changes, invasive species, unsustainable soil management practices. Unsustainable soil management practices in terms of agriculture is that you should be doing conservative tillage. Why conservative tillage we are talking about? Tillage means you are mixing of the soil. Once you mix the soil, your soil becomes very aerobic. Once soil becomes aerobic, the oxygen level goes up. Oxygen level goes up means soil organic carbon will get oxidized and converted to CO2. So you are losing organic carbon. So you should not be doing excessive tillage. So that is why they are saying unsustainable soil management practices, and definitely you should not be dumping lot of chemical, undesired chemical, or a typical concentration is required X, but you are using 5X of chemical, and that you are dumping the chemical into your ecosystem. So that we have to now talk in terms of soil health issue, in terms of soil health management. So let's manage the soil health now. For soil health management, when we are talking about soil health, then you are having physical health of soil, chemical health of soil, and the biological health of soil. And biological health of soil is directly proportional to physical and chemical health of the soil. So if you manage properly the biological health of soil, then definitely you are managing the physical and chemical health of soil. By using different chemicals, we are basically impacting in very way the soil health. And the effects are very much there that because of this, we are creating more barren lands, we are losing the fertile lands, and our productivity is going down. So for that, even if you take a country like US, which is very, you can say developed, very much developed, this is 2020 paper. It says that almost 41% of US land is now having pesticide residues. So you can say, and there the regulations are very tight, you cannot use a particular chemical, and if the chemical is used, a particular level has to be used and not avoided at the upper level. In our country, indiscriminate use is there, then you can imagine in what manner the pesticide level we can find in our ecosystem. So this is the biggest challenge which we are looking for, because if our biosphere goes bad, then forget about our society and economy, which looks right now very good, will have big problem in future. So that way, what we have to think now is a climate smart agriculture, where we have to be weather smart, water smart, seed and breed smart, carbon nutrient smart, and institutional or market smart. This we have to do the interventions. See, there are something which is technology, something there are practices, and last thing is the paradigm shift in our behavior. That is very important. If we put all this together, and that's why this university becomes very interesting, where you are talking about complete development of values. Once you talk about values, then only these things will become very important. Otherwise, you cannot manage these kind of factors. And for smart climate and smart agriculture, we require smart soil health management. And for small soil health management, as I said, that soil health directly is proportional to the biological health. And for biological health, managing the diversity and abundance, or soil microbial diversity management, is the key. And for that, if suppose if your diversity is really right, then your nutrient cycling will become very good. If the nutrient cycling is good, then you will be having water storage quality, because with nutrient cycling, you will be developing more and more soil organic matter. If the soil organic matter goes up, that means you are not losing a lot of carbon. That means your GHG emissions are going down. If these are becoming more, diversity is there. Organic matter with the diversity and their exudates, it will be at soil aggregates. When the soil aggregates will go up, biodegradation will go down and water storage quality will go up. So this is all linked together only with diversity management. So if you manage diversity right, you can essentially manage So how to do this in the right way? If you're looking for a plant cycle, the plants, when it grows from the sapling to a bigger tree or a crop plant, what it really does, over a period of time, you have a peripheral community, plant makes some exudates, it secretes the biochemicals. These biochemicals attracts different microorganisms from the periphery. Once they are getting attracted through the signaling molecules, they will become part of the rhizospheric community. If this diversity is really right, abundance is really right, the opportunity to plant to select the right bacteria from peripheral community to make a strong, fertile and supporting rhizospheric community is really right. So considering this point, in high media we have developed a product which essentially supports the peripheral community to decide and through the signaling molecules move towards the rhizospheric community. And that way what we have shown that if really the peripheral community picks up through the signaling molecule the idea to how to develop our rhizospheric community, then we should define this restructuring by soil microbial community with 16SR amplicon library. And what we have done, we have collected the soil sample from the rhizosphere, we have done the amplicon sequencing, and with what we have shown that if this is my control plot, this is a single plot about 4 acre. In 1 acre we kept control, 1 acre we have put cotton, another soybean and another turmeric. We given our nutrient solution which we have designed, and it shows that in control plot, if suppose there are 5,200 dominating species present based on the amplicon sequencing, then they are basically more in cotton, soybean and turmeric, and at the same time we can even discriminate the cotton versus soybean versus turmeric and versus control. That in what manner a plant essentially decides which community should survive and become abundant in its rhizosphere. That kind of research we have done, and that product is now in collaboration with Kenya, Australia and Egypt, we will be selling very soon. This product we have demonstrated with the different crops, and we have shown that in different crops how the productivity is increasing. Besides the point I want to say that Soil management, if you really want to do that, the soil health management is key. For soil health management, the biological property of soil has to be decided by the microbial community. So you have to protect the microbial community and diversity of soil. If you do that right, the soil health management will be taken care. And if it is taken care of, then your GHG emission goes down, plant productivity goes up. So let's now shift to the next parameter, is the environmental health, which is another very crucial parameter when we are talking about managing ecosystems. What really happens, simple thing, everybody knows that we are applying pesticide through the runoffs, it goes to the aquifers, from the aquifers it comes to our drinking water system and finally comes to us. So we should not be doing that. Second thing is in the environmental health is our solid waste. Biggest problem in our country. The observed behavior should be paper, glass, organic and plastic should be separated and should not be going to the one place. And for that, we are trying our paradigm shift. We want to have society to understand that this is a very crucial thing that we should separate, and then only we can manage and survive in this ecosystem. But we don't do that, and the reason of that is that we have a good societies and surviving with the very good dump sites, right? So we have solid waste dump sites. So that's another issue. So if you look at the linkages, industry, mining, agriculture, drainage, they put pressure on air, water, forest and soil. And because of that, these pollutants getting into the ecosystem. So we require control over this also. This is the typical flow where we are saying that our pollutant, whether it is coming from a hospital like your antibiotics or antibiotic resistance genes, or your industry and a household, they all goes to wastewater treatment plant. From there, it should go to water works and you should get the purified water, but that is not the case in our country now to an extent. And that's the biggest challenge we are facing right now. If I take an aquatic environment, which is very common for the pollution to bring chemicals to the human ecosystem, is essentially your sewage treatment plant, industrial discharge, street runoff, precipitation, and landfill leaching. All they lead to bioaccumulation in terms of bioaccumulation first, followed by photodegradation, volatilization, and microbial degradation. If you look for the recent papers, what really we are finding now, the different pesticides, they are toxic, we know that. But their photodegraded products are more toxic than the original pesticide. And that's another challenge we are posing. And all they collectively make the emerging pollutants. These emerging pollutants are essentially different kind of chemicals in the ecosystem, either through biodegradation or through biotransformation or through photooxidation. They are making the new species of chemical into the ecosystem. And we have no control or even no idea what kind of different pool of chemistry is surviving in the ecosystem. We need to first understand that chemistry and then we talk about different degree of threats. So for emerging pollutants, what really has now shown, at least in marine ecosystem, from soil, water, air, these pollutants are going and they are getting bioaccumulated in animals, plants and microorganisms. And then finally they are getting into humans. For that, very nice study has been reported right now where they are talking about, let me go to that study first. They have shown this study that microplastic essentially is emerging as a biggest threat to the system because there is a lot of bioaccumulation first, then little bit of biotransformation, and then finally it is getting into the human ecosystem in the different pool of pollutants. So that's the another problem which is coming up. So what is the biggest issue right now with the emerging pollutant is that we don't have the risk assessment tools, that how to basically figure out what are the risks. We don't have even tools to measure them. And if we don't have both, then we don't have really the policy recommendations. If we don't have that, then essentially we cannot control the movement of this emerging pollution. And these emerging pollutants with different drivers like climate change and land use continuously leading to biodiversity loss and human risk. So that is the biggest issue right now we are with the emerging pollutants. The another threat is that biodiversity loss means we are losing our forest, we are losing our greens. And this is the typical study NIRI has done, that over a period of time, how Bangalore city has shifted. क्यों भाई? And because of that, you know that what Bangalore city is now facing also, right? So that's the way the new threats are coming. Then this is the new modeling system came up in the One Health or ecosystem health management, where when we talk about modeling, what we really talk about, then that there are different attributes and how these attributes essentially govern each other. Can we have a correlation of different attributes? Can we talk about this attribute becomes a very significant attribute if I apply the principal component analysis. But after doing unsupervised artificial intelligence based modeling, it has been shown that all the parameters, all the attributes are equally supporting the process in terms of going worst. And that's the biggest issue right now. And what the policymakers are saying that let us wait and watch how the things basically evolve. And if this will happen, we won't have any day any solution. Therefore, managing health challenges of the ecosystem will remain as it is if we don't do the properly. Now let's see a few slides what is the coming up or emerging Indian scenario. Indian scenario, we want something like this to that, and we talk about climate is smart cities and so on, that we don't want this, we want something like these kind of cities. To have these kind of cities, first thing we should understand that very soon we will be in the top of the world in generating the solid waste. This is the recent figures, which shows that even we have crossed now US and China. Already solid waste we are generating so much. If we won't recircularize through economy this solid waste, we will be again have biggest issue now with the solid waste. When it comes to municipal sewage waste treatment, we have not more than 30% capacity of sewage waste treatment. That is also a key factor. When we talk about degraded land, almost 1/3 of the land, according to the government record, comes under the degraded land. So we have to restore the land. So land is under problem, right? Sewage water is a threat, and then we are talking about already the solid waste issue. So if we manage that, and finally we have a problem of soil health management because of these three. So we have to collectively talk about this, and then what we have to really do that all these environmental parameters, all the societal parameters and One Health parameters, they have to be discussed on a single platform. If you will discuss these issues in a different or single in isolation, then you will never come up with a strategy to manage in true sense this ecosystem health. Otherwise, we will be talking about zoonotic disease only, but we won't be talking about ecosystem disease. So that is the biggest issue. We have to understand that. And once we will understand that and convert all environmental health, soil health and One Health together on a single platform, then we will be talking about ecosystem health only. So that's the way only we can manage the ecosystem health challenges. Thank you.
Thank you very much, sir, for the wonderful lecture. It was really very informative and knowledge enriching session. So if participants have any question, you can ask to sir. Yes.
Sir, it was a wonderful presentation.
Thank you.
My question is that we are still fighting with many of the electronic waste, because being a human being, we are using so many electronic devices. One person can have so many mobile devices. How can we, as a human being, as a societal person, can take care of that things?
See, the electronic waste is the biggest challenge for any country right now. The problem is that the electronic waste is not a single kind of a metal waste, right? And people are trying to extract the noble metals from the electronic waste, and they are separating the plastic from the remaining of the electronic waste. The processes which essentially looks for separation, they are not cost effective. Therefore, people are not going into that. That is the biggest problem. Cost effective in a sense that if suppose I have this much grams or this much microgram or nanogram of gold present there, its recovery is of no use. So the industry has no interest in converting that into a circular economy. So right now what is really happening, it is converted into big dump, and it goes to the secured landfill after plastic removal, right? But now I think last year the new legislation is coming up or came already, where they are talking about that this waste has to be kept separately. And we expect that in future definitely we will have an option to essentially process this properly, right? See, like in case of mines, you recover gold. Then you have a mine water or mine leachates. Then you talk about microbial leaching and other technologies. They are really good, but up to a particular threshold. If below that threshold the levels of metals or noble metals are there, the recovery is not economical. That is the biggest threat right now. And unfortunately, no society is now responsible enough to say that I don't care for the cost, but I will do that. This is not even happening in the developed world.
So
Yes,
thank
madam.
you very much for an excellent presentation.
Thanks, madam.
Sir, I have one query rather to say. Sir, currently as we are talking about sustainability, but wherever we see, the planet always gets the second place and profit always gets the first place in the development. And even in the society, it is the comfort that people are looking for, the luxury they are looking for. And I think now it is for this generation to really take some very solid initiative if they want themselves or their children living in a better world. So, sir, what can we as an academic institution do? Or what should this generation seriously do to change this situation? Otherwise, Earth is going towards...
Yeah, I agree. See, I can tell you this simple story that whenever there is an industrial development or industrial park comes up or NHI makes the highways, so as per the government rule, the number of trees has been supposed taken off, then accordingly that much amount of money should go to government as a penalty. And that comes under the Campa fund. This Campa fund in India is growing like anything. Anything means you can talk about 1 lakh crores. So that kind of money is there. The idea about that money is to recycle and make the bioresources available back to the society in terms of forestation, in terms of ecosystem development and so on. So government has that kind of plan, right? The first thing. So the required thing is the technology and the development criterias, right? Unfortunately, there are some issues which only government and implementer can talk about. We cannot, an academician can discuss also. So that's a different kind of ball game. The thing you are saying about this generation and so on, they should be given the real picture. They should be told that, see, problem क्या है, हम अपना मोबाइल देते बच्चों को, मॉल में भेजते, right? और एसी कार में ही रखना चाहते, एसी एनवायरनमेंट में ही पढ़ना चाहते हैं. कॉलेज में भी यदि पंखे नहीं चल रहे तो पेरेंट्स आके आपको कंप्लेन कर सकते कि एसी क्यों नहीं लगा देते. सो वी हैव टू टेल देम द रियलिटी, what the way we are abusing our environment, how we are using our resources without really thinking that sustainably how we should be managing. So these reality should be taught. Once you talk about this reality, you can see that even in developed countries, 14-year girl will be shouting in the front of the Senate that you are not managing the environment. Do we see any student shouting for any environmental thing or anything, that why our forests are going or why these developmental activities are coming? Because we are not educating that way. We have to train them, educate them, we have to expose them to the reality. And we have to also understand as a parent and the faculty that what is our responsibility. Till this thing is not came, I will discuss, you will discuss, we will clap, we will say that this is based, but we will never have that kind of based future for our next generation. Right?
Thank you, sir.
Thank you.
Thank you.
Any more questions? Yes, please.
Thank you, sir, for
Yeah,
very
ma'am.
informative session. I just came across one wonderful invention of video. It was related to like liquid tree.
Liquid?
Tree.
Tree.
Huh.
Okay.
So like it, I guess in Serbia or some countries they have developed using microalgae. So like among the like as there are so many like urban this development is so there is no place for trees, so they developed microalgae as a trees
Yeah.
in liquid form, and it is also used to like absorb the your pollution as well as used to generate electricity.
Yeah.
So like I think so they are telling in Delhi also they're going to soon they are it's
Right,
one of
right,
the
right.
most
See,
polluted
the problem
city.
is that people, other country has a spoonful of problem. We have a bucket full of problem. So that's a different kind of management we are talking about, right? But what you are saying is right. The liquid agriculture or
Microalgae they are
microalgae, and this is for the pollution removal. This is for photooxidation, for developing value-added chemicals. This is happening. I mean, just I want to tell you that after next week or so, our team is going to Egypt,
Okay.
where we are talking about hydroponics in 150 acres,
Yeah.
which we don't do that. And this hydroponics we are basically doing using microorganisms, a defined bacteria and defined thing to develop agriculture produce, and we want to have lowest GHG emissions. That is the idea.
Yes. that
So
is
every
also-
society has different needs, and according to that need, we have to manage the problems.
Yes.
Okay, I think one more question is there from, yeah.
Hello. Thank you, sir.
Yes, sir.
Yeah, my name is Kunal. I'm a PhD scholar from VNSU, Surat.
If you can speak loudly.
Yeah.
Okay. Yeah. Sir, my question is like you have a wonderful product from the high media. So I think I remember like, and you are selling it to the other countries only right now currently.
I'm not selling right now to any country right now,
Okay.
not even in India. We are doing all demonstration projects right now
Okay.
because we wanted to be very sure that what we developed is really right and good.
Yeah.
And
Because
it should not be damaging the soil microbial diversity, right?
Okay.
In India right now, we are having 165 places where our trials are going on. And these are the different countries also, they showed interest to do the trials.
Okay, because I thought it like in you are giving your product to the other countries.
Definitely
That's why
not. I'm not going to do that.
No,
I'm
no,
not
no.
going to
I'm
do
not telling. I'm not blaming you. I'm just blaming the system.
Right.
Because major time it's happened like हमारे country में क्या है कि lot of like a system is very like an complicated. So if we are having any kind of product or like in, let's say fertilizer, because we are having a lot of agriculture universities also. So they are developing wonderful work, but why they are not converting into the product?
See, देखो product जो रहता है ना, that is finally comes to the acceptability of the farmers.
Yeah.
So what people generally do, they make product and they want to sell. At High Media, we decided that we are going to make the product, but we are not going to sell for a year or two.
Okay.
We will be only doing demonstration.
Okay.
We want the farmers, the user community to have credibility developed. Once they will have confidence that this product is really good,
Okay.
and when they will demand, that day we will start selling that.
So here again, small input like the farmers are also using antibiotics.
Antibiotics for which crop?
Yeah, they are using for the. They don't know this, but they are getting from somewhere, because I am the AMR inside local brand ambassador. So I am just spreading the awareness for the. They should not use the antibiotics, but still they are getting from the antibiotics from somewhere.
Antibiotics.
Yeah.
Madhvi madam, you want to say in agriculture you are getting AMRs?
Yeah.
I think she is the right person to talk about AMR and agriculture.
Okay.
Thank you so much, sir. So basically, under Gujarat Biotechnology Research Center, we have initiated a couple of programs. One of the program is national or the network program on antimicrobial resistance, superbugs and One Health. And under that, I think I would like to add that 22 medical colleges, 18 veterinary clinics, and several universities who are working on the environmental sites, they have joined hands, and GBRC is kind of a nodal repository, okay, where we are storing these AMR bugs. So far, we have stored more than 9,000 isolates at GBRC, completed the whole genome sequencing of close to 2,500 microbes as per WHO priority pathogen list. And one of the myth, sorry for my voice, it's not keeping well, but one of the myth that we think is, that we use a lot of antimicrobials or antibiotics for rearing animals, and those are creating problems and challenges because ultimately they transfer to human and create AMR. But so far, so if you look at the prevalence of AMR isolates in clinical samples vis-a-vis animal samples, the animal samples have a relatively lower abundance of the AMR isolates. So that is the one myth that I wanted to just bust over here. Another important study which we did as a part of One Health Poultry Hub, which is funded by UKRI, that is UK Research and Innovation, under Global Challenges Research Fund, which is GCRF. So in that we studied that because poultry is reared for the consumption as a food and it is grown in a highly intensified manner. So in that, they are actually fed antibiotics as a part of their growth promotion activities. And in that, if you see, usually everybody sitting in this hall, including myself, will think that we are not affected because we are vegetarian, right? But the poultry waste or the poultry litter is used as a manure in agricultural field. And when you do that, the AMR that is present in the poultry litter is also getting transmitted to soil and from soil ultimately to the plant. So we have proved this with the field studies as well as the pot studies. So these are some of the challenges you need to keep in mind when we want to talk about how AMR is transmitting across food chains, across value systems and across our environment. Sir, I hope I answered your question.
Yeah. See, that's why I said that you should talk to Madhvi madam. The first primary thing I want to tell you that, see, when you say AMR is an antimicrobial resistance gene, gene means what finally? Ultimately, it's some kind of a phenotypic activity. Either it is an enzymatic activity or it is a transport system, right? So if suppose I can tell you that I joined NEERI in 1993, that time I have given a project that I should look for biodegradation of aromatic compounds, particularly the compounds which are heterocyclics or having more rings like phenanthrene, anthracene and so on. As a microbiologist and as a molecular biology person, what I thought first, I got a soil sample. I treated the soil sample with tetracycline because the tetracycline structure. Enrich that sample for 3 months with the tetracycline. I got every single bacteria which can degrade any kind of aromatic. So in environment, this AMR, we are giving a definition to that. They are existing since ages. The day we have crude oil into our ecosystem, from the same day we have AMR genes coming up into our ecosystem. What we are doing is that we are giving them the new definitions. And to be me, as a molecular biology person, I can tell you, AMR, even if you put a pesticide, again a heterocyclic molecule, if a microorganism can degrade that, it can give that genetic determinant to your pathogenic bacteria, and that will become a AMR gene. So AMR is like, you will be always fighting with AMR forever. Yeah, thanks. Yeah, sure, sir. Sure, sir. No, big question is also fine. Horizontal transfer of gene, this is actually AMR is not that big threat. The horizontal gene transfer is a big threat. I can show you that we have published a paper almost 12 years back where we have shown the gene which is for the degradation. And with almost 80% homology, how this gene transfers from the bacteria till the plants with 80% homology. This is we have shown. And this is all horizontal gene transfer. So this is the biggest threat. I can tell you that. See, if you are doing metagenomics also, so you know that we have 80% to 90% microbial diversity not known. Right? And what we really do, we do the sequencing. we do the annotation, and after annotation we say it is undefined, unclassified bacteria, or we say this is defined or classified bacteria. But when we are trying to identify something which is unidentified, and we are annotating, let us say we say this is this kind of AMR gene, I am telling you, you are giving a wrong annotation. Because when it comes to microbial diversity, even 2 base difference changes the species, right? And here with 10% change in homology, you are concluding that we found this gene. Biggest misinformation creation or data creation by us. So abhi we are very, very naive about defining the ecosystem and defining the ecological diversity and defining the AMRs. We have hundreds and hundreds of AMR which we really don't know. And they are the real, you say, the ancestor lineages which creates this kind of genes. Not what we see, the genes which is really there. Vancomycin resistance, this is very obvious. You take a 2 ml, 5 ml culture, put this antibiotic, you will find in 3 days, 4 days, a gene coming up that. So do you think you created AMR there? No. AMR is like a stable genotype which is ancestrally linkages through linked, and it is part of your genotype and only comes by horizontal gene transfer. So you are very pertinent.
Thank you, sir.
Thank you. Yeah, welcome.
I think there is no more questions, right? Okay. Thank you very much, sir. I think
Thank
this is great going, and it is a very good start to this conference because the discussion is very important part and it is going really well. Thank you very much, sir. I request Dr. Raksha Bavankar madam to felicitate Dr. Purohit sir. Please, madam. Thank you, sir. Okay, so going next, the next speaker we have with us for special address is Professor Dayanand Akhzar, Vice Chancellor of Gulbarga University, Karnataka. Professor Akhzar has published over 85 research papers in reputed international journals. His research interest majorly focuses on actinomycetes, bioprocess, and nanobiotechnology. He has completed major research projects from agencies, funding agencies such as UGC, CSIR, and DBT. He has mentored 22 doctoral students. He has visited countries such as Finland, Switzerland, and Czech Republic as an invited speaker, and he is recipient of Hargobind Khurana Best Scientist Award. I request Professor Akhzar to please come on the stage and deliver his special address on diversity and potential prospects of actinobacteria from limestone quarries. Please, sir.
Hello. Hello. Acha. This is the pointer, no? This is the pointer. This is the front. This is the back. Yeah, fine. Thank you. Good morning to all of you. My heartfelt pranams to Swamijis for their selflessness, service to the society, particularly through education. As you all know, education is a business in the Western countries. But if you look back into the education system and the education heritage of India, education is a service. The sort in the true spirit is being carried out by Swamijis, serving the society selflessly through education. Let me thank organizers for having inviting me on this occasion to share a bit of research that is being carried out for the past 20 years or so. That's from 2003 onwards till I'll be sharing with you a research that is being carried out in our laboratory for the past 20 years or so. And before I start presenting my topic, I wish to share with you all that Gujarat state, or the state of the Gujarat, is well known as entrepreneurial state, a business state. And I hope all the young minds in Gujarat will be also quite entrepreneurial in their future. I want to just draw your attention towards entrepreneurial microorganisms. Try to look into the kind of microorganisms that are there in the nature, and few of them are highly potential, highly entrepreneurial. Being entrepreneurial individuals, I want you to look into the very entrepreneurial microorganisms, how you can explore them for the benefit of the mankind. Then only the very basic purpose of the education would be served. Very distinguished speakers, faculties, and the delegates. I would try to restrict myself the diversity of actinobacteria or actinomycetes, instead of talking on the wide aspect of the diversity of the microorganisms. I think each one of us realize this, the importance of microorganisms or the role of microorganisms. Normally, I don't miss sharing the two statements made by a very famous microbial ecologist, Martin Alexander and James Stanley. Their statements makes us to realize how important the microorganisms are. I don't want to read them, and just to put you across about 6 to 7 various major groups of microorganisms. Then biodiversity, very basic information that you can look into any of the text, any of the literature that is available. And what I want to say that in the entire globe, in the entire world, there are few countries which have been recognized as mega diverse countries as far as the biodiversity is considered. Biodiversity, I mean not just microorganisms, it is for the plants, animals, and the microbial community too. And I'll be focusing on the microbial diversity in the environment. Probably Professor Hemant has made my general introduction much effectively, because he was talking about the health scenario, the challenges, particularly focusing the soil health from the point of the microbial communities. So the words which have been put in green, I hope all of you are quite familiar with that. I don't want to explain you habitat, ecosystem, microenvironment and microbial diversity. Until unless you are very, very distinctive from the perspectives of these words, it's very difficult to understand the basics of microbial ecology and the microbial diversity. Please try to understand and distinguish what habitat means, what ecosystem means, what a microenvironment is, and based on all these things, we are trying to understand the microbial diversity. And I'll be focusing the microbial diversity for the region which is well established, right from the state of the Gujarat. If you move towards the southern part, passing through Rajasthan, Maharashtra, Karnataka, Telangana and Andhra Pradesh, along the western Ghats. So we get a typical belt called as Deccan Trap. If you understand the Deccan Trap, the trap is a very unique mineral-rich deposited rocks that have been laid out since ages. And this starts from the Gujarat, right? And go down. And please remember, all these traps are highly variable from one state to another state, or I would say that from one geographical condition to another geographical conditions. By and large, they are quite rich with the limestone with which all these deposits have been constituted. And if you can see these yellow spots on the entire globe, those yellow spots reveals the geographical regions where these kind of sedimentary rocks are quite rich, because of which large-scale cement industries have been established in and around those geographical regions. So I'm just trying to link the limestone as a raw material for the production of cement, how the cement industries are coming up. And then from these geographical rich sedimentary rocks coming to the industries for the cement production, then I'll be touching upon the microbial diversity in those geographical areas, the status, the fate of these microbial communities, particularly the actinobacterial diversity in these particular areas, and I'm looking for quite entrepreneurial actinobacteria amongst the entire microbial community. That's what I wanted to share with you in the next 15 to 20 minutes or so, and then exploring them for their various bioactive molecules, for the welfare of the human health, and then at the end I'll be sharing with you the important aspects, how it can be taken up further. Friends, you are quite aware that now the trend with the young minds is that we want the results very immediately. And whatever we plan, we wanted to complete it to the earliest, get the degree, get the award, get the publications, get the patents, get lost somewhere else without understanding the microbial diversity. It may not be true. It may not be true. But I want to share with you, amongst the microorganisms, whether it's bacteria, fungi, algae, protozoa, viruses, whatsoever it is, then why bacteria are more important? Conventionally, these have been referred as actinomyces to begin with. Then later, they have been referred as actinobacteria. Now we have another unique group, what is being referred as rare actinobacteria. In what way actinobacteria are very significant, are very important when it is being compared with the other microorganisms? If you look back into the literature, we get yourself across the globe, how many institutions, how many groups of scientists or the R&D groups are working with actinobacteria? Not even a dozen. Not even a dozen. It's a very tough task to work with actinobacteria for a simple reason that so easily they will not be grown. You know the growth rate and the multiplication timing of the Escherichia coli. And the actinobacteria takes weeks together to grow and then continue with their physiological aspects. The environmental conditions, the cultural conditions which are required for the actinobacteria are much different from that of the other organisms. So it is from that point of view, normally many are not working with the exploration of actinobacteria. Yes, we have started way back during 2001 or 2, where just incidentally we started working with the actinobacteria. We are not interested, like others, to work with actinobacteria. We started working with the microbial organisms for the production of biocolors. That's how research starts for the actinobacteria. And before I take into that, let me give you quickly what are the actinobacteria which makes them very potential. Unique group of prokaryotic organisms, dual nature of both bacteria and fungi. Very basic, please remember this. This is the only group which has got potentials of both bacteria as well as fungi. And then greater metabolic diversity. You put them anywhere, irrespective of the temperature, pH, alkaline conditions, they do grow. That is just because of the greater metabolic diversity. Higher adaptability to extreme conditions. 45, 50, 70, 100 degrees centigrade. They are quite adaptable, not only to the temperature, many other conditions also. High pH, higher alkaline conditions, and higher salt conditions also. That's how the actinobacteria become so adaptable to the extreme climatic conditions. And it is just because of high GC content, more than 55%, with large genome of about 8 MB. You just compare yourself, what is the normal genome size of the other organisms, you can compare yourself with the Escherichia coli. And because of that, unrivaled capacity to produce variety of secondary metabolites. If you look back into the history of the Sales-Waxman, the production of antibiotics, now you will understand what are the secondary metabolites that are being produced by actinobacteria. They are the promising source of wide range of important enzymes. Recently they have been explored as entrepreneurial microorganisms for the production of enzymes. That is the kind of the research that is being carried out. Relatively less explored for other than antibiotics. Actinobacteria have never been explored other than the antibiotics. Now, my entire research, what we are trying to explore is, we are explore the actinobacteria for other than the antibiotics. That's what just I wanted to share with you at this point of time. Before we look into the isolation of actinobacteria, actinomycetes, yes, I will have to take you to some of the habitats of Deccan trap. I hope many of you are aware of these kind of the geographical conditions. We normally call them as quarries. The Deccan trap, which has got a lot of limestone rocks, after these rocks have been explored for the production of cement, huge quarries left behind in those particular geographical locations since hundreds of years. So these are some of the pictorial presentations that you can see the various limestone quarries. This is particularly in my place, what is being called as the north-eastern part of Karnataka, where our district is located, the Gulbarga, presently which is being called as Kalburgi. In and around we have large cement industries, and those cement industries are digging the geographical region and exploring the limestones, and the quarries left behind are being referred as limestone quarries. You can see their colors, very typical differences in their colors. It is just because of the variations in their mineral deposits within their rocks. You can see this young researcher who has started his career way back in 2021. So I will show you at the end slide, presently who is heading the NCM division of CSR NCL Pune. It is he who has started working with the actinobacteria right from the 2001, and till today our lab is exclusively working with the actinobacteria. So they are some of the students who are visited the various limestone quarries, collecting the samples for the isolation of the actinobacteria. Please remember, as I told you already, to begin with we are never interested for the actinobacteria. We are interested only in collection of the samples, try to explore them for the production of very typical biocolors. That was the basic PhD topic of that particular candidate. So keeping that in mind, we started our research, and it is being ended up entirely in a different way. So there are many students, and this particular slide you can see, not only the hard rocks, we do have a lot of lime powder deposits in some areas of these geographical conditions. It is something like lime powder only. So naturally, these limestone quarries and the lime powder deposits gives a very, very unique ecophysiological conditions for the survival of the microbial communities in general and the actinobacterial community in particular. So we continued with these collection of soil samples. If you look into these spots, it is just partly going down to the Telangana and the state of Andhra Pradesh. Hope Shashikala madam, you will agree with me that there are a lot of locations in the part of Telangana and Andhra Pradesh where we have these limestone quarries. Please try to differentiate the colors which are there in the natural geographical conditions. A typical yellowish color, brown color, grey color, white color, they all makes the difference because of the variations in their mineral deposits in those particular rocks. This is the one I would like to share with you. On spot you can see this, a simple pH paper strips, put a drop of water, put a little soil from the nature itself, and then try to look into the variation in the pH. I hope you know how pH can be tested and how it can be distinguished based on the colors. Please look into the kind of the variations that is there in the geographical nature itself in those lime deposits. Highly alkaline in nature, high pH conditions. You just imagine what would have been the role of organisms and their adaptability for such extreme ecophysiological conditions in nature. That's what just I wanted to show you, and there are the various samples which have been collected from about 17 research students. As of now, 17 students have worked only with the actinobacteria since 2001. Another still 3 students are working in lab exclusively for the actinobacteria. Remember, I am talking to you as a teacher of microbiology, not as a vice chancellor of Gulbarga University. I love talking to the audience as a teacher in microbiology. Yeah, very basics. Very basics I am just taking for the few slides. It is not that easy to grow the actinobacteria like other bacteria, fungi and the algae. The samples that are being collected need to be really subject for the pre-treatment. Otherwise, it is very difficult to grow the actinobacteria on the specified medias. And it goes under the process of calcium carbonate treatment, phenol treatment, heat treatment to eliminate other organisms. It's what we normally call it as enrichment culture, eliminate other organisms, target for the growth of the specified group of organisms, that is the actinobacteria, and a few of the culture media, SC and TGBA, starch, casein, agar, tyrosine, gelatin, brief extract, agar. They are the important organisms. But if you look back, those who are having some interest in the actinobacteria and their isolation, the universal standard media that are available are in the name of ISP1, ISP2, ISP3. International Streptomyces Project, it has been well derived as the media that are being specified for the growth of the actinobacteria. And then culture conditions, it must be minimum 7 plus. They love higher pH, high alkaline conditions. And incubation temperature is always about 40 degree plus centigrade. So that shows how diverse they are in their adaptability to the extreme climatic conditions. And the incubation period is minimum 6 days. That's why many groups, many scientists, many young minds, they are not interested to work with the actinobacteria, because if we incubate today, tomorrow we want the organisms within 24 to 48 hours, we are interested in our results and complete the process of the research or the laboratory experiments. It is not that easy to look for the actinobacteria. If once you get a mastery over the isolation and cultivation of actinobacteria, which you will be realizing into the next few slides, it would be very interesting how important and entrepreneurial these actinobacteria are. I hope some of you might have seen these slides, even in looking into the literature also. You can see these first 4 slides. As I told you, the student and the lab was interested in isolating the beautiful varieties of color producing microorganisms. Because our interest at the present objective was the biocolor productions from the varieties of the microorganisms. We are looking into collect the samples, process it as per the standard procedures, look for the organisms which are being grown, and we have these kind of highly pigmented organisms. If you look into the nature of the actinobacteria, they are quite typical from the bacteria and the fungi. No doubt, they are small, circulated, wavy, powdery, amorphous. Even if you hit that particular colony with a loop, so easily you cannot pick up. It is so hard. It is so hard. Those who are working with actinobacteria would experience that. And coming to another similar plates on a different media, you can see how minute these colonies are. Recently, this is another group of actinobacteria which are being called as rare actinobacteria, which are rarely present in these deposits, and they have got entirely different physiological potential attributes. So these are the two types of actinobacteria and the rare actinobacteria in which we are looking for this. Pure cultures of actinobacteria. This is the stage where the student has took almost 2 years to come out with these particular pure cultures. 2 years only for the isolation point. And finally, he could select, and these 6 important pure cultures of actinobacteria, as I said, looking only from the point of the pigmentation or the colors, because we were interested in production of biocolors from these organisms, right? You know, it was published. There was one famous journal in Malaysia during that period where Asian Review on Biodiversity, Environment and Conservation. It is popularly called as ARBEC. So that journal has published the diversity of these actinobacteria, particularly Streptomyces, based on only the generic level classification as its cover page photograph. And that publication was attracted by 3 important groups across the world looking for the actinobacteria. And one leading group, what is being referred as father of actinobacteria in China, is Professor Wen Jun Lee. So he's a compound microscope under the electron microscope. Yeah, if once we know their colony characters, if once we know their microscopic features, yes, naturally we are interested looking for their simple biochemical and physiological tests. Please try to remember, Burgess manual gives these are the basic physiological conditions which need to be tested before we identify those particular actinobacteria. I don't have to repeat those things, but they are very basics. Please remember, unlike bacteria and fungi, it is at this level itself, it confirms very clearly the most probable genus to which these particular organisms belongs to. Incidentally, all the isolates that have been got from these particular samples, they belong to one notorious genus of actinobacteria, that is the Streptomyces. I am using very consciously the term notorious genus. Sometimes, many times we use to share with you, notorious students. I hope you understand. And please remember, only those notorious students will be very, very intelligent. They will be very, very intelligent. Only thing is they need to be properly directed for their notorious things to explore further. Remember, among the actinobacteria, the most notorious genus is only the Streptomyces. If you look into any other bioactive molecules that are available across the globe today, it is only because of the genus Streptomyces. And later I will take you to the various species of this particular Streptomyces. That was the stage where naturally student was interested for a publication. Sir, क्यों नहीं हम एक publication कर देंगे? Because already he had a credit for this biocolor producing microorganisms which are being accepted and published in the ARBEC. Then we written the paper, we have submitted for the publication. Nowhere it was accepted. Nowhere it was accepted. I tell you the reason why it has never been accepted. So accepting the publication regarding bacteria, fungi, algae and other things is much easier, particularly when being compared with the actinobacteria. Because to publish a particular bacteria, it is very much essential that these particular isolates need to be evaluated or subject to the examinations at these three levels. That is basic systematics, which had been completed. We wanted to publish and it was never accepted. And then we are compelled to go for molecular systematics. I don't want to explain you what the molecular systematic you mean, and you'll be looking for some more slides now, how exactly these have been subjected for the molecular systematics. Not only to that level, later they were compelled to subject these isolates for the chemosystematics, a kind of a unique chemical component, the content that is present, both quality and quantitative, in the cell wall and the cell membranes of these actinobacteria. So basic, molecular, and then chemosystematics, then only one can think of publishing these particular organisms from the point of the diversity. And please remember, ours was a public university, a government university. The facilities were very, very poor, and our institute was located in a very, very rural area, northeastern part of the Karnataka. Facilities were very poor, except basic equipments. There were nothing in our laboratory, except the strong mindset of the student, who has been already invited by a research institute at China by Wen Jun Lee. He has made, nahi sir, main jata hu wahan par and I'll get these things done. Remember, in the span of about just 18 months, he could give us 6 new species and 1 new genus of the Streptomyces. So what you will be realizing now, that's the kind of the work which is being carried out by that particular candidate, who is a principal scientist in the CSR NCL today, heading NCM division. You will come to know a little later who is he. Right? Now, with that simple molecular analysis, you know that they are the prokaryotes. Organisms need to be tested for the genomic DNA isolation. Then 16S rRNA gene sequencing. I don't want to discuss the standard procedure that was there. You know how difficult it is and what kind of facilities also required to go for that kind of a molecular analysis way back during 2004 or 5. And all those isolates have been definitely tested for the genomic isolation of the DNA, and then PCR amplification of that particular 16S rRNA from that purified genomic DNA, and after that PCR amplification, that 16S rRNA naturally would be gone for the sequencing and construction of phylogenetic tree. So these are the 3 major stages. Each one of you quite aware. If you are not interested, get back yourself into the literature. You will understand how to subject the organisms before they are being identified for the molecular systematics. If once these 3 stages are over, yes, we have to go for the construction of the phylogenetic tree, or the dendron, what is being called. Right? I just quickly take you through all these 6 important phylogenetic trees of these organisms. The sample based on which the coded was this. First one was DAS 131, that was the isolate, and that is the phylogenetic tree. I think the basic concept you are quite aware what exactly you mean by the phylogenetic tree and how genomically it can be related specific with its neighboring organisms. With that percentage of the differences or the similarity, the organism distinguished can be identified. The next one was something like DAS 165. Remember, we had thousands of organisms which have been named with these particular codes, and only those selected 6 have been worked out at that particular research institute. The next is DAS 139T, a specific isolate that was coded in the institute. And then this is the only one organism which was quite different from the Streptomyces, that was Saliphilia from the east. So we could not continue with the work with this particular isolate. And then Nanomyria indica, which is another important new species with that particular phylogenetic tree, and Actinidectinspora indica with that particular strain. Yes, organisms. So we cell wall amino acids in that particular species or the isolate, and you can see there are about 6 species with one standard marker. That's how for the comparison it has been well assessed. Evaluated, and then it will be reported. Evaluated, and then it will be reported. After that, now we have played with the publications. We are ready with the basic systematics, we are ready with the molecular systematics, we are ready with the chemosystematics, and then you may not believe the papers which are submitted earlier for those journals, they were after the authors to finalize the script and submit for the publication, and we could not do so. We started playing with the publishers. Why I am sharing with you is that is the stage where every candidate must reach. You should know how to have authenticity as far as reporting of the new isolates are considered. Then we have selected only 2 to 3 important journals. You know that in the entire world, IJSCM is one standard journal which always accepts only either new species or the new genus. International Journal of Systematic and Evolutionary Microbiology. There is a one important. Then Anthony van Leeuwenhoek from Germany, which is exclusively accepts only the new species or the new genus. So there are hardly about 3 or 4 important journals across the globe which really accepts only the new species and the new genus. We started publishing these articles and submitted to these particular publishers for the publication. And the first novel species is being referred as Streptomyces gulbergensis. I would like to share a bit story behind naming of these organisms. So earlier I hope the professionals are aware that normally we used to name the organisms either by the name of the guide or by the name of the student, or at the most by the name of the institute. Remember, as far as Actinobacteria are considered, there are international set of standards where the naming of the organism should be only in a particular pattern. It can never be named after by the guide or by the student or by the institute. It is only the potential. I was telling you, this organism is so entrepreneurial. Why? I'll tell you later. And that entrepreneurial potential attribute has to be included in its name. And one of such is the habitat or the place from where this organism has been isolated. We thought let credit goes to my place from where this organism has been isolated, and it has been accepted as a novel species in the name of Streptomyces gulbergensis. Gulberg is a place where we have isolated, we have collected the sample, the organism has been named, right? And all these features are well. You can see the molecular phylogenetic tree, the basic biosystematics and cultural conditions, and the important, you can see the phenotypically distinction with all its chemosystematics too. It is not that easy to understand. Anybody can go for the journals, and these slides are, I'm leaving with the organizers for the benefit of the students. You can circulate, so anybody, those who are interested, you can relook into these details. Second one is Streptomyces tight tolerance, physiological potentiality. This is the one species which has been named as tight tolerance because it is highly tolerant to temperature, highly tolerant to pH, highly tolerant to the salt conditions, higher salt conditions. That's how this new species has been published. Streptomyces deccanensis. We moved to Telangana, Andhra Pradesh. As I said, this is the Deccan trap, which is a very, very unique geographical conditions which is being universally accepted as Deccan trap. We have given the credit of this particular species as Deccanensis because one of the soil samples among the Deccan trap has been collected and it has been named. This is the third new species. Fourth one is, this is the first organism which have been isolated from the Indian soils. It was never been reported earlier. Hence, I moved on to the country. The credential were given. So, please remember, these are the established standard criteria for naming the organism based on which we started giving the nomenclature to particular this novel species. That is the Nanomyria indica, the first Nanomyria species that has been isolated from the Indian soils. Allostreptomyces indica, again another genus among the Streptomyces species which has been isolated for the first time from India. So these are the 6 new different species. And surprisingly, we never expected that we would be able to get a new genus from the Indian soils amongst the actinobacteria, that is Actinorectispora indica. It's a novel genus as well as a novel species. Why? A simple basic systematics, molecular systematics, chemosystematics, very typical physiological potential with which these organisms probably are the highly potential from the point of their bioactive molecules. So this was a stage where we had the project from the University Grants Commission and also the DBT. And we wanted to continue this project. This was the stage where student has come back from China, and we wanted to continue this project with the next generation of the students. But DBT has put a major query as far as this is considered. The major query was the basic mandate of the DBT during 2013-14, if I am right, the task force, they asked me a simple question. Why do you want to report the new species and the new genus from the natural conditions? Whether you report or not, it is there in the nature. What is its use to the mankind? When DBT, Government of India, is giving huge amount for the point of the R&D projects, it should serve the purpose of the mankind. Then the mandate from the task force was, don't look for the anymore isolation of the new organisms. Try to explore those novel species and the genus only from the point of the bioactive molecules. Can these organisms produce the new molecules or molecules, bioactive molecules with new characters or the potential which can serve the mankind, either through the environment or through the plant or the agriculture or the health of the humans, whatsoever it is. Somehow, so again we have got another DBT and DST project to explore these organisms for their bioactive molecules, what is being normally we try to put them as bioprospective attributes. Actinomycetes, you know, classical actinomycetes means we try to call them as antibiotic producing organisms. It is not the stage now, absolutely not. And later it is the advanced antibiotics. Diverse metabolites in which we were interested to look for the organisms from the colors point of view. And then enzymes and the pigments. Suggestions given by the task force committee, explore these organisms for the antibiotics, novel antibiotics. That was the only one condition which was not accepted by our team. We have decided not to explore these organisms for the antibiotics anymore. Sufficiently, right from 1950s and 60s, these actinomycetes have been thoroughly explored for the antibiotic production. Why can't we look for other bioactive molecules other than the antibiotics? That was the challenge we presented before the task force. Task force has accepted, and then they have given us a project to explore these novel isolates for the enzymes and the pigments. Our interest was the pigments. Biocolors we have kept as it is, but no pigments would be produced by the organisms without the mediation of the enzymes, catalytes. So that's why we started looking the exploring of these organisms for the enzymes as well as the pigments. The first enzyme we started working with the tyrosinase. It was just a general observation. If you cut the apple, keep it for a while, exposed to the air, it will turn into the brown color. Cut the brinjal, cut the potato. Few of these vegetables or the fruits, if you cut them and keep it exposed for the air, oxygen, slowly they convert themselves into the brown color. Just we are interested in the biocolors. So this is the concept what we have got it from the literature, and we wanted to look for the enzyme involved in the process of this transformation or production of this color. And the enzyme mediated was exclusively tyrosinase. We wanted to explore these organisms for the production of tyrosinase. So that's how our interest has been initiated, exploring these organisms for the tyrosinase enzyme. And these are the details with which you can look at later. How do you screen the actinomycetes for the tyrosinase production? Standardize the medium, give the standard incubation conditions, and then try to look for the kind of the pigmentation. The density of the pigmentation gives you the kind of the enzyme that is being synthesized, that is being produced by the organism. That's how it is being identified. So surface view or a rare view, a dark brown pigment if it is there, it is true that this particular pigment is being produced because of the tyrosinase mediated physiological process, conversion of tyrosine into the melanin mediated by so-called enzyme as tyrosinase. I am taking you to the enzymatic details that you can look back into the basic literatures. Yes, unfortunately or fortunately, all the organisms, novel species, were well known for the production of tyrosinase. Then we started standardizing them from not only from the quality but also from the quantity point of view. A very basic procedures have been followed for screening of these organisms for the production of tyrosinase by streptomyces. You just standardize the whole process, broth, inoculum size, pH, temperature, incubation period. They are very basic things in the bioprocess engineering. Standardize and then try to differentiate. An important software-based method which is being used for the quantitative estimation of the production of any enzyme, that is RSM, response surface methodology, is a software-based methodology which automatically gets itself integrated with all the media components, standardize itself for the maximum production of the tyrosinase enzyme. And that has been done, and we could get a tremendous increase, significant increase in the tyrosinase enzyme after process variables and then after critical variables as far as RSM is considered. Just I am showing you a few more slides only from the point of the application point of view. One of the important application that was developed by another student in the laboratory is this tyrosinase is very important, which catalyzes the polyphenols, monophenol, diphenol and the polyphenol. Testing of the phenols in the natural compounds, whether it is in the oil or whether it is in the water or the soil. So it is something like, take a small piece of the filter paper, put the tyrosinase enzyme, put the different water samples or the soil samples with variations in the phenolic contents. The kind of the pigmentation or the density which is highly varying out of that particular strips on which you can name what kind of a phenol it is. It is something like our target was try to make use of this tyrosinase in detection of the phenol pollutants in the natural samples of water and the soil. So that was with this particular intention where this work has been done and it has been published in IJAB. And I don't want to continue with that particular any other enzymes, just I am shifting to the pigment, that is the melanin. I hope all of you are aware of the melanin. What is melanin? You don't have to tell me. You just try to understand what exactly melanin means. Right? So, the natural pigment which is present in all living beings, particularly the human beings, plants and the animals. The colors, because of it's only the melanin, which is a natural pigment that is present on the skin surface. Right? The skin. Right? I don't want to get into the details of it. Just I would like to take your attention towards the biological functions of melanin. Look into all these various features. That is the kind of the biological role that melanin is being played in various organisms. Physical strength, which gives to this cockroach, you know, it is not that easy to hit the cockroach. Its surface is so strong and that color is because of the melanin. So melanin, based on its functional features, functional components which has got on its structures, right? So it plays a different biological roles in different animals and the living beings. And you can see this thermoregulatory also, and it is a defensive against predators. And you can see this proper functioning of the site also, the site with which you can just locate. You just surprisingly look at the biological functions or the attributes that are being taken care by the melanin. So we felt that yes, melanin does have a significant role where these organisms can be explored for the melanin production instead of just tyrosinase. So that's how our interest started with looking our exploration of these novel species for the melanin point of view. That is the application point of view. I think you know this melanin protects against UV-induced damages, sun protection factor, cosmetics, sunscreen lotions. I think I don't want to explain particularly the girls, right? And incidentally, we see a lot of girl students in the life sciences. So the case in particularly with the microbiology. Remember, it is the melanin which is a very, very significant chemical component which act as surface protectant agent. Now, we have got an idea how we can explore this particular melanin that is being produced by the organisms in the cosmetic industries. That's how the applied aspects has started as per the mandate of the DBT, and then we have started working on it. Not only to the surface protectant factor, we thought of exploring this as a dye to the hair and also the textile. You know the fashion colors? Hair dye? What kind of the colors? You just imagine, whether in Western or the Indian conditions. Please remember, we are interested just to, why can't we apply this particular natural melanin from the, as an hair dye? This is a kind of a project what we have with the IICT, Indian Institute of Chemical Technology, CSR laboratory in Hyderabad. The work is going on that particular line. Only those two things are sufficient. Started as per the standard procedures, screening of streptomyces for production of melanin. Again, increase the production of the melanin by same RSM method. Like tyrosinase, we have standardized the process for the pigment production. And this is the one stage where I wanted to redefine the research. What is the research? For my understanding, not from your point of view. With a particular character of the student. Now I am coming to that point. Now, was students what we normally do? Because I have also passed through that stage. When we isolate the cultures, do all the things, and cultures need to be, the slants, whatever that has been left into the sink, they need to be thoroughly autoclaved and washed and cleared, and then take up the next experiment. So this was the one stage where that student had been to his native place for almost for about 45 days. He could not clean the test tubes where there were hundreds of cultures. He has dumped in the sink and he has left to his place. By the time he has come back to the laboratory, it was almost for 45 days. Of course, as usual, nicely he got the scoldings from the guide that was going on. But please remember, a kind of insight that student had, which has made him today to lead the NCM division as a head. He had an entirely different mindset to observe those cultures. After 45 days, the normal surface cultures of the actinobacteria were entirely changed into a different observations. The surface, which was with a particular pigment, had developed some kind of a droplets on the surface of the culture. It is something like a droplets of gum. So those cultures took almost 45 days to exude kind of droplets on the surface of the slant cultures. Then I started asking, sir, this is something different. To begin with, we thought probably they are the water droplets because they are dumped in the sink. No, it was entirely different. That is the curiosity which has started. That's why I said the results were entirely different, which was not expected. And simply, that student, we have planned in such a way that use micropipette, suck that particular droplet and then test it for these particular TLC, UV absorption and FTIR. The basic tests that are essentially required for the analysis of any of the bioactive molecule. We thought probably there is a bioactive molecule externally synthesized by the organism that has come true. And that was a pure melanin produced by the organism. Pure melanin produced by the organisms. Then it was a surprise for us. Is it so? When we look back into the literature, some of you might be aware that earlier the detection of antibiotics happened in the same way. Who has discovered the antibiotics? And his basic observation was a different pigmented droplets on the surface of the cultures, which was led into the observations of the antibiotics when they have been tested against different pathogens. In the similar line, melanin droplets have been tested. The next important issue, a very big news in the Hindu news. You know that any breakthroughs definitely will be covered by the news and the media. What is important was how is that these droplets of melanin could be produced by the actinobacteria like antibiotics. That has a major boost for us to submit another project to the DBT and get the fundings. That's how our research has initiated for the production of this melanin. And not only that, please remember, his negligence of not washing the cultures led him to a patent. If he had washed the cultures, he would have gone, then patent would not have been come. What I am trying to tell you is, sometimes our features also help us, provided if we have a third eye. Third eye. Only your two physical eyes are not sufficient. You must have a third eye, a different mind to observe very, very clearly your progress of the cultures and in the process of the research. That has led into a patent of production of water-soluble melanin. The melanin that is being used in all the cosmetic industries, in the synthetic industries, is only water-insoluble melanin. It is a high, costly affair to use this water-insoluble synthetic melanin in the production of the cosmetics. Now that is the patent what we are given. This was the 13th patent in the world wherein we could report the extra synthesis of the production of water-soluble melanin from this particular organism. Now it has led into the different applications further. Bacterial mediated melanin, then slowly one of the students started working with the more quantitative production of bacterial melanin also from the Pseudomonas, again which was isolated from the same limestone quarries and the lime powder deposits. I quickly go through the slides and its analysis, confirmation as a water-soluble melanin, and then its antioxidant activity, yes, and then UVB protectant activity. This is the very typical character that has been attributed to the melanin. It protects all the UV rays, particularly the UVB protection. So it has been tested against a particular cancer cell lines also, where this melanin has been proved. Water-soluble melanin that is being produced by this Pseudomonas and the Streptomyces has been well proved as UVB protectant bioactive molecule. This is the kind of work that is being carried out in the IICT, which is being shared with the industries for the production of cosmetics using this particular water-soluble melanin. I cannot reveal further. Right? But anyway, you can look into these particular. So you can go for these particular publications. An important publication which has been come out of it, those who are interested in understanding. Why I am sharing with you at this stage is, Gujarat is known for the Deccan Trap. Gujarat is known for very, very unique geographical conditions. Those who are interested, not only actinobacteria, even for bacteria and fungi, if you are interested, get back yourself to the nature, collect the samples, have a third eye, try to isolate the organism, explore them for the bioactive molecules. You being the highly entrepreneurial individuals, as I made at the beginning, so you can look for highly entrepreneurial microorganisms. Really, you'll be serving the society. You'll be serving the society with a great revenue, Ultimately, entrepreneurial means what? Getting more money, generating high revenue. Yes, this is the way how you can think of being microbiologist. You can look for entrepreneurial organism too. This is a kind of thing what I thought yesterday I have added when I asked Mosami to prepare this slide. Mosami is my laboratory student. She has worked with these things, Dr. Mosami Das. I asked her to just, today morning I called and I told her to prepare this slide so that I can share with particularly the professionals or the teaching faculties also, young teaching faculties. Try to have a projects in this line. Look for the microbial diversity and bioprocess technology as a title like, and then you can look for the regional and the national microbial diversity. Try to explore the regional biogeographical conditions and the national biogeographical conditions where you can collect the samples, screen them for bioactive molecules, not necessarily the enzymes and the pigmentations. Anything that you can look even for the antibiotics too. And then try to explore them from the application point of view against not just human health. You can go for the plant health and also animal health. The keynote address which was given, the health of the animal, health of the plant, health of the human being are most important. actinomyces can be the very entrepreneurial organisms to give you hundreds of bioactive molecules which can be proved in the industries of health industries, agriculture industries, and also the veterinary industries. That's how you can look for the revenue generation. And then the microbial nanotechnology. I have not covered any slides because these bioactive molecules are clubbed with the nanoparticles that have been synthesized by these organisms itself, which are getting more efficient in their applications point of view. That's how the microbiology department, incidentally, we have got a very good faculties. I'm told that there are about 13 faculties in the Department of Micro, and majority of them are young. The second advantage is highly diverse faculties. So try to integrate. Strength is highly diverse. Vision is to be how to integrate and then try to explore the microbial diversity to serve the humankind. That's how just I thought of adding this slide today morning to share with them. Development of bioprocess and enhanced production of L-glutaminase by Streptomyces rochi. So you can just, those who are interested, you can talk to Dr. Mosmi. It is her research which has been carried out in my laboratory. Just I wanted to focus you this particular slide. It is she who has carried out this particular work. Recently, we had an interactions with one of the UK universities, University of Bradford, where the bioactive molecules from the Streptomyces and one of the plant species have been explored against particularly the human health. You know how difficult it is, the curing of the wounds in the diabetic patients, what we normally call them as the diabetic ulcers. In diabetic cases, ulcers cannot be cured so easily. So we had a kind of collaborations with the University of Bradford and our university, wherein bioactive molecule from our laboratories and the nanoparticles from their laboratories are being used in electrospinning technology. There are only 3 institutions across the globe which are working with this particular technology. How exactly these two, bioactive molecule and the nanoparticles, can be spun together, make it as a layer. If it is can be applied to the ulcers on the wounds of the diabetic cases, that can be cured. There's a kind of the product development project collaborations we have with University of Bradford. Recently we have, this is the collaboration, and then we have organized a conference on that particular line where this is the professor from our laboratory and then professor from University of Bradford. These two are the experts from bioactive molecules and the electrospinning technology, where we are looking for a particular product also in this particular line. And then recently we had organized a joint collaboration conference with that University of Bradford, where 13 country speakers were attended this particular conference, which was held in the last month at our university during January 8th and 9th. What I'm trying to share with you is, those who are interested in your national and international collaborations, only thing is it requires very, very hard work and a third eye to open up differently from the others. Sky is the limit, those who are interested in your career. Of course, their visit, there's a kind of their visit, and I was telling you that our research was in collaborations with the IICT, Indian Institute of Chemical Technology at Hyderabad, where these molecules, both tyrosinase and the pigment, is being explored as a dye as well as the cosmetic agents. You might have heard these two scientists, Professor Ashok Pandey, who was earlier in NIST, now he is in Lucknow. So he is the mentor of mine and my team to put me on the line of the research of the actinobacteria. He was the first man in during 2001, encouraged me to take up the work on actinobacteria, otherwise which we never interested in that particular. And it is Dr. Syedji Dasgir, the first slide what I have shown during 2001, he was the student of my lab collecting the samples for the looking for biocolors production, and incidentally all things have been turned out to be the actinobacteria, and hence the way forward in my lab even today, producing about 17 PhDs only on actinobacteria and their bioactive molecules, working with 4 more students. Of course, based on that expertise, we have visited different countries, exploration of the actinobacteria. Yeah, you can see, he is Professor Wenjun Lee, what I said, father of actinobacteria, who is in China right now. You may not believe, or it will be surprised if I say that he has already reported 177 new species and 42 new genera of Streptomyces. There's a kind of the command he has got over the actinobacteria. Those who are interested, you can contact him. For grand sky is the limit. I'm using the term, for grand sky is the limit. Only thing is you must have a potential organisms in your hand and you must have a potential mind to explore them for the welfare of the mankind. That's my team whenever we visit the slide. So I put it normally as ART, Actinobacillus Research Team. Thank you very much for your patient listening. I'm happy if you have any other queries also. I think I've taken more time.
No, it is. Thank you very much, sir, for really a motivating lecture. And I think all must be agree that sir has rightly explained a small observation can lead us to breakthrough research. Thank you very much, sir. If any questions are there, you are welcome to ask.
Oh, yeah, madam, please.
So thank you for the excellent presentation and sharing your work with us. Sir, I have one query that except Allostreptomyces indica, all other novel species had a very huge range of pH tolerance. So this pH was whether affecting their morphology or their ability to produce a particular metabolite, or was it in this whole range they were functional?
Madam, let me share with you. It affects both. Morphology, yes, no doubt about it.
Yes.
That's why all its sporulation and mycelial features based on the temperature and the pH.
Yes.
They are the two important issues,
Yes.
physiological conditions which affect normally any microorganisms. The most important thing is these extreme environmental conditions also makes these organisms for the production of unique extremolites. Now a recent concept is we don't only say the metabolites, we call them as extremolites. A kind of metabolites that are being synthesized by the organisms in extreme physiological conditions. That's how it is only the actinomycetes which are getting for the production or which are being explored for the production of extremolites. Extremolites particularly in the cosmetic industries.
One another thing that that 45 days vacation that your student had gone for reminded me of Louis Pasteur when he developed chicken cholera. Similarly, he had gone for the vacation and came back. So to have a third eye is really very, very rare.
No.
Thank you, sir.
Now that's what I just wanted to share with the students, so that just mere interest is not sufficient. Just your hard work is not sufficient. You must have a very unique observations. That's why I said a third eye is must. Everyone cannot do that. Thank you, ma'am.
Sir, it was a wonderful presentation on actinomycetes. I have a question that you have somewhere shown us the rare colonies of the actinomycetes. Can you just give more insight on that? How can we work upon it, or what are the characteristics that they possess?
I'm sorry, get yourself to the website of NCL Pune. Contact Dr. Syed Dastgir. He would continue that work. We have not initiated that work, but it is another potential group with a very, very micro colonies which have been universally accepted as rare actinobacteria. Literature reveals that rare actinobacteria are much more potential than the actinobacteria as far as their extremolites are considered.
And there could be a chance of getting them only to those particular locations that you have shown in.
Not necessarily, not necessarily. But you know, these are the conditions which right from Gujarat, they throw the Deccan trap, you can look into it. So that can be a very good project, you can think on that.
Okay. Thank you, sir.
Thank you.
Any other question?
Anybody?
Please stand up.
You are the mic.
Yeah, okay. Yeah.
Yes, namaste sir. Thank you for the beautiful presentation. As I myself working on actinomycetes, so it was very thoughtful and in detail presentation. Sir, specifically I would like to know about role of actinomycetes towards the pollution degradation. As you explained about the enzyme production industrially, but I would like to know if you can tell me about the role of actinomycetes towards the pollution degradation, specifically plastic degradation or hydrocarbon degradation, if you can, sir.
Hydrocarbon degradation again is being taken care by the Dr. Syed Ji Dastgir.
Okay, sir.
Because I've been already superannuated 3 years back. No more I'm going to the research labs. Only the remaining students are working with that, with only the enzymes and the pigments. That's why I said it only gives you a big gateway for exploring the bioactive molecules by the actinobacteria. You can do that.
Thank you, sir.
And I must, sir, you'll be surprised if I say that 72% of the revenue of NCL is being generated by this particular Professor A.G. Dastgir. You just imagine what kind of revenue can be generated from the microorganisms. 72% of the entire revenue of the NCL Pune, of the CSIR NCL Pune, is being generated only by this particular laboratory, exploring the organisms for their bioactive molecules. Yes, please.
Okay. Good afternoon, sir. It was very informative lecture. My question to you is that these days when we get to expose to new knowledge, information and topics, everything fascinates us. So how do you decide what is your topic of interest to work upon and research upon?
You have to decide on yourself first. What is your interest? What is your ability? The second aspect is, which is more important relatively? Everything is important. Relatively, which is more important, in which you are interested. So I told you, neither the student nor my lab was interested in taking up the actinomycetes work. It was just incidental. We started with the looking for the organisms for the production of biocolors. They turned out to be all actinobacteria. And then it was attracted towards the Venjunli. And then our whole research has been directed in a different way. So what is important is, you have to thoroughly understand the literature that is being existing from the point of the future perspectives. So based on the interest that in which you are focusing, you can decide yourself. And the best is, let me share with the guides, my colleagues, please remember, it is the role of the teacher which is most important to trap the talent of the students, which is the right area, which is the right perspective for the students to undertake the work. So the students should be at that particular stage. It is the job of the teachers.
Thank you, sir.
Thank you.
I think there is one more question there. Yeah.
I think that would be the last question. If any more questions, we can discuss during the lunch hours.
Thank you, sir. It is really very understanding and informative session. Sir, I want to ask you that, is any database or any data set of biomolecule produced by the extremophiles?
Yeah, very much available.
Please tell me.
Yes, it's very much available. Go to the websites, you will get it. Okay. Extremolites from actinobacteria or extremolites from the extremophilic microorganism.
Okay.
It is only the latest concept. That's what is being explored. Looking for the bioactive molecules from the extremophilic organism. Extremophilic means it is a natural ecophysiological conditions. Temperature, pH, salt concentration and the sugar tolerance.
Okay, sir. Thank you, sir.
Thank you.
Thank you very much. Sir, if you allow me, I have also a last question.
It's my pleasure. Please.
Sir, when I was doing my PhD, our lab was also working on halophilic actinomycetes. And what we found that usually we do pre-treatment. And in that pre-treatment, we used to get the same kind of genera and species. So don't you think that we should try other media as well, and without pre-treatment, then maybe we can get newer ones or maybe rare ones?
Okay, the first point I would like to share with you is, absolutely there is no literature. Nutritional conditions, nutritional components of the medium, you can look for a specific organism.
Okay, sir. Thank you, sir. Thank you so much. Thank you, sir. Now I request Dr. Mosmi Das, madam, to please felicitate Professor Agsar.
A student felicitating teacher. That's a great award to a teacher. Thank you. Please. Thank you so much. All the best. Thank you.
Thank you very much. Thank you, sir.
It will take a lot of time.
No, sir. It was perfect. So participants, with this we declare the end of the first technical session. Now it is time for a lunch break. And the venue for lunch is Aswad Canteen. The volunteers will escort you to the place and venue. Thank you so much.